This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 766, recorded on June 8th, 2021. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Ann Arbor, Michigan, Kathy Spindler.
Hi, everybody. Here it's 84 degrees Fahrenheit, which must be about 29 Celsius.
Wow.
Guess.
It's 32 Celsius here.
Yeah, 29. Oh, it says it's raining, but it's not really. This is the Norwegian weather app, so yeah, there must be a little delay and it's wrong. Okay.
Also joining us from Madison, New Jersey, Brianne Barker.
Hi. Unsurprisingly, it is the same weather here as it is with Vincent. It is 90 Fahrenheit, 32 Celsius. Kind of gray. Looks like it's getting ready to rain.
Yeah, I just got a radar notification. Heavy rain starting in 15 minutes. Okay. Hopefully the power will stay on. We have a guest today who is coming to us from Philadelphia, Pennsylvania, the University of Pennsylvania, David Fajgenbaum. Welcome.
Vincent, thanks so much for having me on. I don't know the temperature outside because I haven't ventured outside all day, but I can tell you that it is very dark. So I think there's some storm clouds heading your way.
So I say your name right, Fajgenbaum?
Fajgenbaum, you nailed it.
So I heard other people say it before. That's how I knew it. Otherwise, I wouldn't have known it. So let me just tell you about what David's positions are. So he's a professor of medicine. And he is the founding director for the Center for Cytokine Storm Treatment and Laboratory Research. And boy, what a great department. Cytokine Storm Treatment. Love it. And he's the associate director for the Orphan Disease Center, and finally co-founder and executive director of the Castleman Disease Collaborative Network. Did I have in there the COVID site? You're also involved in that. It's not on the list, huh? Okay, so we're going to talk about the Corona Project, which is the main reason I asked you to join us today. And you also started that up as well. Is that right?
That's right. I launched the Corona Project last March.
And that's what I heard you talk about before, and I thought that would be cool. But since then, when I asked you to join us, you sent us a copy of your book. So David has written two books, one published in 2015, We Get It, and then one published in 2019, Chasing My Cure. You know, the 3-word titles. It's good. It's good, right? It's only bested by Stuart Firestein as a Columbia professor. He's written 2 books. The first was called Failure, and I can't remember what the second was called.
I believe the other one is called Ignorance.
Ignorance.
Nice.
And he said to me,
titles.
And I had him twice on TWiV to talk about it. He said, I don't know what I'm going to do for my next book. I can't think of another one title. Anyway, David, we'll talk about the Corona Project. But first, let's get a little bit of history. And tell us where you're from and your training and so forth.
Sure. So I grew up in Raleigh, North Carolina, and went to college at Georgetown, where I actually played college football there. I was a quarterback at Georgetown, and that was kind of a lifelong dream. And shortly after I got there, my life really changed when when my mom was diagnosed with brain cancer. And it was her diagnosis with cancer that really refocused my life from this dream of playing football to now wanting to dedicate my life towards medicine and searching for treatments for patients like my mom. So I went to grad school at Oxford and then went to Penn for medical school. And as a medical student, a third-year med student, doing my training to hopefully treat cancer patients and be involved in research, I became the patient myself. I became critically ill with a disease that was not diagnosed at the time, eventually diagnosed as idiopathic multicentric Castleman disease, where my liver, my kidneys, my bone marrow, my heart, and my lungs began to shut down. So I was hospitalized and spent nearly 6 months hospitalized in the intensive care unit with multi-organ failure. Eventually got the diagnosis, and with the diagnosis came treatment. But I was so sick when I was finally diagnosed. I actually had my last rites read to me, which of course I know you know from reading Chasing My Cure. But thankfully, with the diagnosis, I got multi-agent chemotherapy. It saved my life. I was able to return to medical school, but unfortunately, I went on to have a number of relapses and realized that I couldn't continue my old life. I had to search and chase after a cure.
At what point did you decide to write the book?
I wrote the book. I decided in 2017. So I, as you know, and I guess we're kind of giving away the ending to the book, but the fact that I'm on the show gives it away. I'm alive today, but I nearly died 5 times in a 3.5-year period after my diagnosis, multi-organ failure, months in the hospital, and I ran out of options. There were no more drugs in development. There were no more promising leads. And so that's when I went to work. I started a foundation called the Castleman Disease Collaborative Network. I began conducting laboratory research, and I eventually found a drug that is literally saving my life. And I started taking that drug in 2014. It had been around for decades, but never used for my disease. And in 2017, the New York Times ran a story called "Doctor Cure Thyself," and that actually was one of the title ideas for the book. But this "Doctor Cure Thyself" article really highlighted the effort to find a drug that was saving my life. And I received a lot of feedback from people saying, like, this story needs to be shared, in particular because it highlights the importance of drug repurposing and the potential for really incredibly shortened timelines between patients who are really suffering from terrible illnesses and then finding solutions. And of course, I really wanted to share the lessons that I learned from my journey, lessons that have really fundamentally changed who I am.
Yeah, it's a terrific book, David. It's beautifully written, and I mean, I'm just amazed. It is great. I recommend everyone read it. Chasing My Cure.
It's riveting. I'm only about 2/3 of the way through, and that's because I stayed up late and got up early. As soon as I get home, I'm going to finish it tonight.
Yeah, no, really fantastic. Really fantastic. I'm only about halfway through myself, but great so far. So one thing that you mentioned was that you realized that you sort of had to change your old way of life a little bit. And I can imagine some of the trainees who might be listening might wonder sort of how you kind of finished medical school through all of this. Can you talk about that a little bit?
Sure. So I first became sick as a 3rd-year medical student. I spent a year on medical leave, came back, and I was on this experimental drug that we all hoped was going to work for me. And so back to med school, kind of back to the grind. And then I relapsed on that drug before I finished my 3rd year of med school. And to your point, that's when I realized that I couldn't just kind of keep going back to the grind, like, ICU, back to med school, and that I would need to dedicate my life to studying Castleman disease. And so when I got out of the hospital after that month, that's when I started conducting laboratory research into Castleman disease, basically in free time during medical school, and started this foundation called the Castleman Disease Collaborative Network. And for me, I knew I wanted to finish medical school. And I also around that time realized that I thought that I would need to go to business school to actually pick up some of the skills that I thought were really some of the rate limiting steps to make progress for Castleman disease. I realized the technology was out there, the science, the medicines were out there, but harnessing those resources and making sure that we were all moving in a common path forward felt maybe like the most important thing I could do. So I finished medical school, went on to do an MBA, in part because I knew that I wouldn't be able to do a rigorous internal medicine residency like I'd always wanted to do if I was back and forth to the hospital. And so I actually finished up my MBA and then joined the faculty at Penn in a research track position where I could focus my career 100% on research. So though I have an MD, I really I'm on a track that's really all of my peers are PhDs.
So it seems like this CDCN, I wrote down the initials and now I can't remember, Castleman Disease Collaborative Network, changed the paradigm for how research on orphan diseases are investigated. And I wonder if you could talk a little about that.
Sure. So as you're probably well aware, the traditional way that rare disease research occurs is that groups will raise money and then invite researchers to apply to use that money how they see fit. You basically hope that the right researcher applies for the right project at the right time. And that works really well if there are dozens of applicants and billions of dollars, say the NIH approach. But that doesn't work very well if you're a rare disease and you have 3 researchers in the whole world. What is the likelihood that one of those 3 researchers is going to have the best idea and also the skill set to actually drive forward on that idea? So we came up with a really simple solution, but as you said, it's really fundamentally changed the field. And that was, we said, well, maybe rather than raising money and then hoping the right person applies at the right time, what if we turn to our community of physicians, researchers, and patients and actually ask them, what are the important research questions that need to be answered? What are the studies that could be done to answer those questions? And then we would go out and figure out the best people in the world to actually do that research. And so we went out to people and asked, we said, this is prioritized. You're the best person in the world. Will you do this study? And sometimes the first question I would get is, what's Castleman disease? And I'd go, oh gosh, this isn't a good start, right? I want you to do Castleman disease research and you're asking me what it is. But that, for us, that was immaterial because we just wanted to figure out what's the most important study and then who's the best person in the world to do it. They didn't need to necessarily know anything there was to know about Castleman's.
Well, and didn't you also say that when you first got your diagnosis, you quickly Googled it yourself? So
I did. Yeah, I was a third year med student. And the first thing I did was Google it. And actually my doctor was out of town and his nurse practitioner actually was the one who delivered the diagnosis. And when she delivered it to me, she said, I don't know anything about this disease. So if you want to learn about it, you need to Google it. And that's, for me, that was fundamentally such a challenge as a medical student where I'm in this medical system where you feel like we have all the answers to all diseases and that we've got drugs and solutions. And all of a sudden I realized, oh my gosh, there are diseases out there where we don't know a thing about them. We don't know how to treat them. We don't know how to diagnose them. And Castleman's isn't the only one. There are 7,000 rare diseases.
I have to say, I did know what Castleman's disease was only because it can be caused by human herpes virus 8, which was discovered at Columbia by Chang and Moore. And they always used to say, among the things it causes is Castleman's. And okay, multicentric. I don't know what that means, but so
that's right. So one of the forms is caused by HHV8 infection. And most of those patients are actually co-infected with HIV virus. And so that's one group. And then there's this other group that we call idiopathic multicentric, where you get the same cytokine storm, the same symptoms, but you can't find a virus and we don't know what triggers it.
Okay, so Castleman's is a cytokine storm disease, correct?
That's exactly right.
But we don't know what the trigger is, right?
So in some people, it's HHV8. In some people, it's a malignancy. You can have actually a monoclonal plasma cell population that triggers the cytokine storm. And then there's this third group, which we call idiopathic, because we can't find a malignancy, we can't find a virus, and we just get really, really sick with a cytokine storm.
So I know that there have been some immunologists, especially during the COVID pandemic, who have started fighting against the use of the term cytokine storm.
Yes.
So I guess, what are your feelings on that, and kind of what specifically are you describing as you're describing the cytokine storm?
Yeah, it's a great question. So I totally agree with all of those people who feel that cytokine storm is not appropriate to be used to describe COVID-19 for the vast majority of COVID-19 patients. Of course, most patients are asymptomatic, and many that are symptomatic or mildly symptomatic, they are not experiencing a cytokine storm. I think that the only appropriate use of cytokine storm are the most severely ill patients. But even in that category, it's hard to say someone has a cytokine storm versus doesn't if there is no definition. And so I had the real honor of working with Carl June this past year and writing a review that was published in the New England Journal of Medicine, trying to define what is a cytokine storm. Because again, you can't really argue if something is something or isn't if there isn't a definition. And so we established a definition, which I really encourage any listeners or viewers to reach out to us if you have any suggestions for improvement. But basically, you need to have elevated systemic levels of cytokines. We don't define which ones, but elevated circulating cytokines. You need to have acute inflammatory symptoms, which you see with these conditions. And the third one that's tricky is you need to have organ dysfunction secondary to an overexuberant immune response. And so the point here is that if it's idiopathic multicentric Castleman disease and there's no virus, then any sort of organ dysfunction is excessive organ dysfunction due to excessive inflammation. But if you have a virus like SARS-CoV-2 and you have a mild amount of organ dysfunction, but you mount an appropriate response and you get better, I don't think that describes a cytokine storm. It's those patients where there's excessive inflammation. I think the best way to assess is there excessive inflammation is to look at randomized controlled trials and to see, do patients get better if you use something like corticosteroids? Do they get better if you use something like baricitinib or tocilizumab? And if so, at least those patients, the ones who benefit from it, those patients likely had excessive inflammation and should be called cytokine storm.
this the one aspect that you mentioned in the book, blood moles, do you think that's going to be a diagnostic feature?
It's actually in the diagnostic criteria for Castleman disease. This eruption of hemangiomas, which is a marker of excessive VEGF levels.
Huh. So how many people get Castleman's globally? Do we know?
So the numbers aren't very clear, as it usually is the case for rare diseases. Within the U.S., using insurance claims databases, the estimate is about 5,000 new diagnoses per year. So quite rare, but not what you might call ultra rare. Out of those 5,000 cases, about 1/3 of them have this idiopathic multicentric Castleman disease subtype that I have.
Then has anyone done genetic linkage studies to look at that?
Not enough genetic work has been done. In fact, we actually started out by first saying if some forms of MCD are caused by a virus, let's search for a virus. So we worked with Ian Lipkin, who I know is at Columbia as well. We worked with Earl Robertson at Penn, working with Danny Dewick down at NIH to look for potential pathogens that could be causing idiopathic multicentric Castleman disease. That's where we focused most of our attention early on. We have not uncovered any either novel or existing pathogens. We thought we might find HHV9, but we did not find HHV9, unfortunately, because it would be great to know the etiology. So we focused heavily on an infectious trigger. Thus far, 3 studies have come back negative. It's always hard to prove a negative. At the same time, in parallel, we focused a lot of attention on phenotyping the disease. So doing serum proteomics, flow cytometry, single cell RNA sequencing to understand, even if we don't know why the cytokine storm is starting, let's just figure out what the cytokine storm entails. And then we can think about therapeutic options. And that's where we identified sirolimus, in my case, as being a treatment approach we wanted to try for me. And now we're using it in other patients. And now we're finally getting to do more genetic work.
And the other name for sirolimus is rapamycin?
Rapamycin, that's right. Yes, which has such a long and interesting history. Of course, I wrote about it in the book. Such a fascinating molecule discovered in the island of Rapa Nui, Easter Island, as a bacteria produced in the soil or in the soil of Easter Island. Really amazing story.
So are there a particular group of cytokines that tend to be seen in Castleman's disease patients?
Yes, interleukin-6 is the characteristic cytokine. In fact, you all, I'm sure, very familiar, and everyone, or many listening, are familiar with Tocilizumab, which blocks the interleukin-6 receptor. It has been approved in the U.S. for many years for rheumatoid arthritis, but actually the history of Tocilizumab is that it was developed by a friend and colleague of mine in Japan for Castleman disease, for idiopathic multicentric Castleman disease. And I share this story in the book of Kazu. He discovered that interleukin-6 was elevated in Castleman's patients, and he went on to develop a drug that blocks the receptor Tocilizumab. But before he gave it to patients with Castleman disease, he decided to test it on himself. He gave himself Tocilizumab. And when I asked him about it, I said, Kazu, Makoto just told me that you gave yourself Tocilizumab as the first patient. He said, no, I didn't give it to myself. The nurse, she gave it to me. I said, exactly, Kazu. So he discovered the target. He developed the drug. He gave it to himself as the first human. He didn't die, so it went on to get tested in other Japanese Castleman's patients, and it got approved in Japan for Castleman's, came to the U.S. for RA, and of course, you all, I'm certain, have seen the use of Tocilizumab in the setting of COVID.
But you got Toci, right? And it didn't help, correct?
That's right. I did. And so for about 1/3 to almost a half of patients, IL-6 blockade with Toci or Cytokine is effective. But for the remainder of us, a little bit over 50%, IL-6 blockade is not effective. And that's where we typically get multi-agent chemotherapy. Until now, sirolimus is an option for some of us.
What do you think about the efficacy of Toci? So one is a receptor blocker, and the other is the ligand blocker. What is your feel about the value of that for COVID?
It's a really good question. And as you know, and as we discussed briefly, I launched the Corona Project because early on in the pandemic, I observed, as many others did, that there were a lot of questions like this. What drug looks to be effective? What drug might be effective, not effective? And I realized that we needed to have a central database of all of the drugs being tried for COVID, and then to systematically assess which drugs have the strongest evidence and the least strong evidence. Within the Corona Project, which has aggregated this data, Toci one of the more challenging drugs to pinpoint whether it's working and who it works in. So as we're all aware, one of the biggest challenges in figuring out drugs that are going to be effective for COVID is that it's already been clear, particularly dexamethasone, but in other cases, the drugs that are effective at one time point are oftentimes not effective at others and can actually be harmful if given at another time point. So there's heterogeneity within the same patient, depending on the timing of when they get it. And then we also know there's heterogeneity between patients, that some patients are more likely to benefit from one type of drug versus another. Some patients do not have an overexuberant immune response. Other patients do. So the complexity is really hard to deal with. But the totality of evidence, in my opinion, is that tocilizumab does seem to have a small effect, but a real effect in patients if given within 24 hours of admission to the intensive care unit, not if given very early and not if given too late. It's really kind of that Goldilocks perfect time frame.
And so I presume it's your experience with Castleman's that it led you to found both the Cytokine Storm Center and Corona, right?
That's right. Yeah. So I experienced a cytokine storm. I nearly died from one 5 times. I found a drug that's been at my neighborhood pharmacy for all these years that's saving my life. And so when I joined the faculty at Penn, I decided I wanted to dedicate my career to doing similar work, profiling cytokine storm disorders, and then searching for existing drugs that could potentially be repurposed to treat those cytokine storm disorders. That's the central thesis. And then the third piece is then systematically tracking whether those drugs actually work or don't work. So really a 3-step process. And so all of a sudden, and we launched this center about 4 years ago, certainly well before COVID. And of course, at Penn, there's a strong interest in cytokine storm in the setting of cellular immunotherapy, because a lot of patients with cancer that get cellular immunotherapy will develop a cytokine storm like we see in Castleman's and COVID. And so running this center, I found myself on Friday, the 13th of March, 2020, which I'm sure you all will remember very well, was the day that the US shut down. And I found myself that evening thinking to myself, I really hope that some researcher somewhere will follow the path that we went down to find a drug, in my case, sirolimus, that could be helpful. We'll follow the path of looking at all FDA-approved drugs and figuring out what might be repurposed. We'll systematically track it the way that we do for Castleman disease. Gosh, I really wish there was a center out there that could do that. And then about a minute later, I was like, wait a minute, why am I waiting and hoping and wishing that someone, somewhere, some other group does it? Maybe we should just do it. If I had waited for some researcher or some center to figure out a drug for me, I wouldn't be here today. And so anyway, I turned to my team that Monday morning and we all agreed that we were going to launch the Corona project and try to add some data, or at least some structure to the data around drugs for COVID-19.
So where are the data that you're using coming from?
So a few different sources. The primary source is PubMed. So as you're well aware, there are literally, now it's thousands a week. Back then, there were about 2,000 papers published in the first 3 months of COVID, but there was a large body of literature emerging on drugs being used anecdotally. We didn't know how many drugs were being used, but we knew that they were showing up in the literature. So we launched this project where we assembled a team of medical students from Penn and from other med schools to just go paper by paper through the about 2,000 papers. And we thought, maybe there's a couple dozen drugs that are being tried for COVID. And amazingly, we found in the first 9,152 patients that over 100 different drugs were given to the first 9,000 COVID patients. So doctors were trying lots of stuff. Just like, let's just try this, let's try that. All kinds of things were tried. And so that made us say, okay, well, if the first 9,000 patients, 100 drugs have been tried, maybe we need to keep going and keep tracking this. And so PubMed is the primary data source. We also work with a group at the FDA called CureID. They have an app that if you're a provider and you treat patients, whether it's with COVID or some other infectious disease, and you ever use a drug that is not approved for that use, go to the app, enter it in. It's a way to keep track of these off-label uses of drugs. So we use CureID data as well. And I hope all of you will consider taking a couple minutes to enter data in CureID. But now we're looking to expand beyond just published data, which is the primary data source, but really and I should mention now there are over 500 drugs that have been used in the first about 270,000 COVID patients that we have data for in our database. So now we're looking to use insights from the 500 plus drugs to then look in EHR databases, insurance claims databases, to see if we can generate some hypotheses we can test out in those other data sets.
So Corona is only repurposed drugs, correct?
No, we do include new drugs as well. So the N at the end of Corona is new.
All right.
So we do include new, but really we started out focused on repurpose because of course the first 3 months of the pandemic there were no new drugs tested at that stage.
So it's basically a database, and you can go in and search for whatever drug you're looking for, correct?
Yeah, the idea is that you could either go to PubMed and read 29,000 papers, or you can go to cdc.org/corona and you can play with the data. All 29,000 papers worth of data are there. And so, and we'll, if you type in Tocilizumab, you can see that 4 out of 10 randomized controlled trials have achieved their primary endpoint. And you can go to the 6 that didn't achieve their primary endpoint. You can see how close were they to being a positive trial, how far were they, how many trials are currently underway, the number of patients on these drugs. So yeah, I highly recommend go to the website, play with the data. Don't read 29,000 papers unless you want to assemble an army of 100 medical students to do it.
So it sounded like at first this was really just a volunteer, make use of people's time while they're shut down and not doing their medical school stuff and et cetera. But you've recently gotten some funding to expand this. Can you tell a little about what that'll allow you to do?
That's exactly right. So last summer, I had a number of conversations with officials from HHS, FDA, and NIH who said, this is great. We're using your data because, again, we don't want to read at that stage. It was probably 10,000 papers. We don't want to all read the 10,000 papers. So it's great that you've got this database that we're utilizing. We hope you'll continue to grow it, and there may be an opportunity for federal funding to support the continued growth. And so we really doubled down because we were so thrilled that the federal government was utilizing this data. I mean, that's why you do things like this, right? Is to be able to support this global pandemic, and the fact that the US government was using it. In particular, they were using it to pick drugs to go into clinical trials. So they could use this data to say, okay, what are the drugs that need to be in the next RCT? So that was really exciting. We really doubled down in our effort. And federal funding often has slow turnaround, and we're still waiting for our notification. I don't know if it'll ever come. But in the meantime, and a wonderful nonprofit, the Parker Institute for Cancer Immunotherapy, which of course does not have a focus on COVID, but they really were impressed by the work we were doing, and they wanted to contribute and support the work we were doing. So we got a grant from the Parker Institute for Cancer Immunotherapy to actually be able to hire data analysts to go through these papers instead of relying on unpaid medical students from all over the country. And so that's been really wonderful to be able to have the boost of being able to have some full-time employees working on it.
So David, what are some of the winners in the Corona database, in your opinion?
Yeah, so the obvious winner at the top, of course, is dexamethasone. We spotted dexamethasone really early in the pandemic, and we thought we thought there was a mistake because there was only one recommendation that the whole world was following, and that was don't give corticosteroids to anyone. There was no what you should give to patients. It was just don't give corticosteroids. That was the only recommendation. And we're getting piles and piles of data through the Corona Project on corticosteroids being given to patients all over the world, and anecdotal reports of them helping. And we're just like, what is happening here? Only one recommendation yet. So anyway, of course, we all know the way that the story ended, and that's that dexamethasone is very helpful if given late in the disease course. A clear winner. Tocilizumab is certainly in the mixed category. Baricitinib is in the early category. It's an immunosuppressant that has rationale for why it might be helpful, but it certainly is still early on. We've got about 10 drugs that fall into this promising but not yet proven category. We give school grades like A's through F's to drugs, and I think it's helpful. And so anyone can go to the website and see how we grade things. And it's all based on data. It's an algorithm that's fully informed by data. It's not any one of our favorite drugs. But importantly, some of those promising drugs that I'm particularly optimistic about, they actually were the drugs that the Corona database was utilized by. I was part of the Active 6 working group that selected drugs for the Active 6 trial, which is now going to be the US's largest COVID trial once enrollment ramps up, over a $100 million study. So some of the drugs we picked from the Corona database for Active 6 that we're particularly optimistic about, and inhaled budesonide is certainly something that we're very optimistic about early on, if given at the right time. Fluvoxamine early on in a disease course shows promise. We also are optimistic about fluvoxamine later on in the disease course. Thus far, there haven't been any RCTs of late stage fluvoxamine, but based on MOA, it seems certainly plausible. And so it's really, for us, it's about focusing on randomized controlled trial data, and particularly the very small ones. There might be a trial of 30 patients, 50, 60 patients that cannot definitively tell us that a drug works, but they can give us a signal. And so something like intravenous immunoglobulin has had a number of RCTs, very small, 30 to 50 patients, but suggesting a mortality benefit. And when you have a trial of 30 to 50 patients and you see a mortality benefit, certainly could be a false positive, but it should get your attention. And for us, our goal is to look for those, we call them diamonds in the rough. They're drugs that show a positive signal, small trial. So most people kind of write them off and say it's a small study, it's 30 patients, 60 patients. But our job, we feel, is to pick up those diamonds in the rough, take them to the NIH, take them to the FDA, and say, we've got to do a larger study. I'm not saying that they work, but I'm saying that if we have to invest in, and we can't do 500 RCTs, right? It'd be great if we could, but we can't. And so if we have to pick, let's pick from the ones that look like the diamonds in the rough.
How does convalescent serum fare, in your opinion?
It doesn't fare very well in our database. And part of the problem, so I'll just tell you, objectively, it has a D. And the reason is that only 1 out of the 7 RCTs that have been published have shown a benefit. However, and I know that I feel like everyone always has a however anytime you talk about any drug in COVID, there's always like this, wishy-washy. It may not be totally the case, but I will say with convalescent plasma that the trials that have been done have tended to be later on in the disease course. They've tended to be hospitalized patients, ICU patients, people already on oxygen. And so, one would suspect, at least based on the monoclonal antibody data that we've seen with Regeneron, Eli Lilly antibodies, the earlier you give, the more effective they are. And so one could infer that convalescent plasma, though it's only, I think, 1 out of 8 or 1 out of 9 in terms of being effective or meeting a primary endpoint in all of the trials done, it has not been studied extensively early on in the disease course. So that's one of the challenges we face. We can only use the data that are available to us. The data available to us are not very promising, but we haven't looked at it from all angles.
We get a lot of questions about ivermectin. Maybe you could give us your thoughts on that one.
Gosh, I said tocilizumab was a tough one, but ivermectin I think is even more tough than tocilizumab. And part of the reason for that is that at least with tocilizumab, there are at least 3 studies that have over 1,000 patients in each of those studies that were done really rigorously and have turned out to be positive, which really helps you to lean towards thinking that it's helpful. Ivermectin has had more randomized controlled trials completed than just about any drug other than hydroxychloroquine. I think it's number 2 in terms of completed RCTs, but almost all of them are very small. And the ones that were large have had some notable concerns in both directions. So there were some positive trials where there were methodological concerns, and there were some negative trials of methodological concerns. In fact, one of the large negative trials, there's suspicion that a large number of the patients in the placebo group also got ivermectin. So now if many of your placebo patients are getting your treatment arm, then obviously you're not going to be able to have a nice comparison. And so I think the tough thing with ivermectin, and I've been getting a lot of emails recently about ivermectin, and what I respond is just the listing of all the trials that have been completed. And if you look at them, they've split about 50-50 between looks like it might be helpful, looks like there's no benefit. And so this is a tough one. I think that I just recently heard from some officials in Trinidad and Tobago in the Caribbean, and they were asking, what do you think about mass ivermectin use? And I think that the tough thing is the data does not support that it is effective. But in the middle of, tough times where basically no one in the population is vaccinated right now, and they are really struggling, you have to ask the question, does the fact that there have been randomized controlled trials that have shown a benefit, is that enough for you to infer that maybe there is some benefit of the drug?
I think you said on a call that I was on that what we need is a 4,000 patient RCT for ivermectin,
yes,
right?
100%. And the good news is it's coming. So Active 6, Active 6, I don't think it's been fully announced yet, but I would be shocked. So I can't guarantee it'll be an Active 6, but I would be shocked if it's not an Active 6.
That would be great. And then, but, despite the outcome, people will still use it either way, right? Especially because it's so cheap and, when dosed properly, it's pretty harmless. So it's hard to argue against it, right?
That's right. And, there's been a lot of arguments early on that the dosing and the levels that you can get in sera are probably not, maybe not the levels needed to have the really strong antiviral effect. So there's a lot of debate. And to your point, it just needs a really large trial. And then we just need to be able to believe the trial, because having a lot of kind of mixed results is really, really tough. And I should say that I cannot give any sort of an official stance that ivermectin will or will not be in Active 6. But I would suspect that it will be in Active 6.
Sure.
I want to go to something that's a little more back to the Castleman's disease story. And that's, it sounded like it took 11 weeks before you got your diagnosis. And then when you got to Arkansas, the shuttle driver looked at you and said, "Oh, you're here for Castleman." And it's like, yeah, so if it takes the medical profession that long, but being familiar with it increases the probability that you can figure it out. But what is the normal time to diagnosis maybe for Castleman's? And then what are the challenges in trying to figure out or to reduce the time to diagnosis for such rare diseases?
Yeah, this is a great question. And exactly when I arrived in Little Rock, Arkansas, which is where the world's expert for Castleman disease is, just as you said, the shuttle bus driver saw me and recognized in Castleman's patients, we had tremendous fluid accumulation all over our body because of this multi-organ failure. And so I had a huge belly and I had fluid just kind of everywhere. I had about 40 or 50 pounds of fluid all over my body. And you're right. He looked at me and he said, "Oh, you've got Castleman disease. You're here to see, Dr. Van Ree." I was like, "Oh my gosh, I've been at all these major medical centers and no one can diagnose me." But here we are. So part of the problem back then, which I'm so thrilled we've been able to make some progress, is that there was no diagnostic criteria back in 2010 when I became ill, meaning that for a lot of rare diseases like Castleman's, there are clinicians who know what to look for. Pathologists are aware of what it looks like under the microscope, but no sort of consensus has ever been agreed upon or established among experts in the community. And so there was no criteria, so there was no checklist that anyone could go to. And at one point it was suspected, but you need a lymph node biopsy. And so it took 11 weeks for that biopsy to be performed. But one of the first things we did through the Castleman Disease Collaborative Network was to bring together a network of physicians and researchers and patients, importantly, from around the world to establish the first ever diagnostic criteria. And so we established that and published that in 2017 in the journal Blood. And that was a huge step forward. So one is just to get people to agree to it. But then two, as you know, in the medical community, getting an article like that into Blood is so important because then it's going to be read by hematologists around the world. So that was step one, establish it, get it out to the world. Step two is also to raise general awareness. Chasing My Cure actually was this great way of getting the word out about Castleman disease to a lot of people. A lot of doctors will email me and say, I read your book, and then this patient showed up and sounded just like you, and we went through it, and the patient's got Castleman's. And so that's really, really exciting to hear that there's awareness being raised. But I think that the third piece that we really need to get to is we really need a solid, consistent, positive diagnostic biomarker. We need a blood test or a stain that can be done that's very reliable. Right now, the diagnosis of Castleman disease requires a doctor to cut out your lymph node and then to look at the lymph node under the microscope. And it needs to look kind of like lymphoma, but not be lymphoma is basically what you have to do. And so, it would be much better if it could be, you need to look kind of like lymphoma, not be lymphoma, and have this stain of this level that's this positive. And then you know that you've got IMCD. So we're working on that third piece.
You mentioned lymph nodes. I think in the book at one point you went in the freezer and took out some of your sections to look at them again, right?
Even better, Vincent, it wasn't even just that I went to the freezer. It was that during my relapse, I asked my doctor to cut the lymph node out because I knew that if I survived, I wanted to then be able to go into the freezer to get it. But first I had to actually, in the middle of the relapse, say, will you cut out a lymph node? And there's a clinical justification because Castleman's can turn into lymphoma. And the only way you know that is if you cut a lymph node out. And so there's clinical justification. But for me, it was all about the research justification was that if I survive this, I want to have some tissue to work on after I get out of the hospital. And so, it was only in the freezer because, I had asked for it to be taken out.
I just wondered when I read that, how often do clinicians, scientists, when they get a disease, take such an active role? I'm not sure it's all that common, right?
I don't think it's all that common. I think there were a lot of factors that contributed to kind of the perfect storm for this. I mean, first off, I'd say I was very reluctant early on. I didn't want to be like a physician scientist searching after my disease. I just wanted a doctor to tell me what to take and just like get better. And then I kept relapsing, kept being like, someone's got to have an answer for me. And by the fourth time I was like, oh gosh, okay, I guess I need to try to like, basically channel whatever remaining time I have towards this, knowing really, all in well that I likely wasn't going to find anything that was going to be helpful for me, maybe someone else in the future. So it was very reluctant, but I will say that also the timing of where I was in my training as a third to fourth year medical student, that's when you have some free time. And I had some wonderful mentors. Arthur Rubenstein was and is this incredible mentor to me. And so I think a lot of things came together to make me feel like I wanted to spend the time doing it, and then also to support me in actually making progress.
Although when you first were admitted, I remember you started asking questions and the doc said, you have to be the patient.
Yes.
I'm the doc. And you said, he said it sternly, but he was probably right.
Yeah, at the time I knew he was right. And it was like, okay, I know, but I want to be the patient, but you're not figuring this out. So like, I want to be the patient, I want you to be the doctor, but figure it out. And at some point, like, if you don't figure it out after a certain amount of time, other people need to come into the picture. And of course, one of those people who came to the picture from the book is Tony Fauci. Mm-hmm. And, this is, of course, well before Corona, but my dad got this phone number from a family friend who was like, you should call this guy. He might have some insights while your son's in ICU without a diagnosis. And so my dad just calls him every day. And for 30, 45 minutes, he talks to him, has no idea who he is, by the way, but he tells, my dad tells him all my labs. And every day for weeks, he just gives my dad advice. And I was like, Dad, who's that guy at the beginning of the hospitalization like 6 months ago? He's like, oh, I don't know. I think his name's Fauci. You should Google him. He works at the NIH. Like he just thinks he's like, an employee of the NIH, right? So I Googled Fauci, NIH, and I learned that it was Tony Fauci. And, of course, he'd already won a Presidential Medal of Freedom. My dad should have known who he was back in 2010. But he wasn't interested in small talk. He was like, my son's got this, and this, and someone told me you can help him.
I also thought it was amazing when you went to see the physician in Little Rock, you brought a PowerPoint stack with you. And he actually looked at all 100 slides, right? Something like that.
He did, and I didn't know that he would, but thankfully he did, because if you go all the way, to Little Rock, Arkansas for an appointment. But no, you're right. It was, I had been meticulous because, as you'll remember from the book, I was in the hospital for 7 weeks without a diagnosis, and I was actually discharged. I just started getting better, and I wasn't satisfied at all with this idea, because I remember my ICU doctor looked at me and he was like, let's just hope it doesn't come back. And I was like, gosh, that's not very satisfying.
Wow.
So yeah, I was busy digging into things, and I could share all that with him.
So this is making me think a little bit about someone I know who has a rare disease. And so I'm wondering kind of what your recommendations are for other patients who kind of are in this situation of having trouble getting a diagnosis. Should they make a PowerPoint slide and have their dad call Tony Fauci?
Yes, and yes. No, I think that number one, it's about reaching out to the expert. And so in my case, it was emailing Fritz Van Ree, and he, agreed to have me come see him. But number one is figuring out who is the expert, and then just getting in your car and just driving until you get to that person. I don't care how far away she is. The world's expert is who you need to go see. So I think number one is go to the world's expert. Number two is that when you're with the world's expert, recognize and understand that just because they're the world's expert doesn't mean that they know all that there is that needs to be known about that disease. The world's expert only knows as much as the world knows, right? And so go to the world's expert, but realize that he or she is not going to have all of the answers, and that it may involve a team-based approach where maybe you're going to see someone else as well. And really, you want to make sure that that person who's your primary treating physician is okay and kind of humble enough to realize that, like, none of us know all the answers, and the only way we can get closer to truth is if we all work together. And then I think the third piece of it is really a central theme throughout my life, but also throughout chasing my cure. And I mentioned I was reluctant to realizing this, but it's the realization that if you're going to hope for something, so it might be a treatment or a cure or a diagnosis, then that should be the thing that drives your action. And so, it's really reflect on, okay, what are you hoping for? And then what can you do today to get closer to that? And realizing that there's going to be a lot of times where you trip and fall and you don't get to where you want to get to, but just to stay persistent.
Your dad interacted with Tony Fauci. Have you had a chance personally to interact with him subsequently?
I have, yeah. I met him in person at NIAID about a year and a half later. I went there on behalf of the Orphan Disease Center at Penn. So Penn got a large grant to start an orphan disease center right around the time I was getting back from my medical leave. And so I met with Dr. Fauci to talk about the Orphan Disease Center. And we had a really, really good laugh picturing my dad, mispronouncing his name, not knowing who he was. And he said, yeah, your dad kept calling me Fucci, but I didn't, want to correct him. And I don't think my dad knew if Fucci was first name or last name. He was just Fucci. And it was just like, this is what's going on. There was no doctor at all. It was just, hey, Fucci, these are the labs, what's going on here? So no, we had a really, really good laugh.
I'm amazed that your dad was able to talk to him every day. That's amazing.
Every day. It's amazing, isn't it? I mean, I guess in 2010, there wasn't as many, things pulling him in a million directions. But I think it really, more than anything, I think it just highlights, Dr. Fauci's just commitment to patients. I mean, it's not part of his job description to answer phone calls about complicated, diagnoses in the ICU at Penn. I mean, that's not. But of course, he's managed to do that and so much more.
And I think a really good doc is always willing to hear something like that, right?
I agree, except every day, for lots of time is pretty special.
Oh, I know. I do a podcast with Daniel Griffin on COVID every week, an infectious disease physician, and sometimes patients send in emails, and he'll say, "Here's my number, just call me."
Wow, that's awesome.
And I mean, a lot of people are listening. They could all call him, right?
Yeah.
I don't know how he does that. I'm sorry to change the subject again, but I wanted to just mention. So I noticed you only go to D on your database. You don't do any Fs, right? Yeah,
we're gonna start having to put some drugs into the F category. Okay. Part of it is about certainty. So we call something an A when we feel quite certain that it's effective. We want to call things an F when we feel quite certain they're ineffective. It's hard to really get to certainty when, as we discussed, every perturbation hasn't been done. But the reason we're going to get more confident in giving an F grade is because we're going to split it into outpatient grade and inpatient grade. And so, for example, we can feel more confident that dexamethasone is an F for outpatient, but we, of course, wouldn't want to call dexamethasone as an A for inpatient,
sure,
right? And hydroxychloroquine, we can feel very confident it's an F for inpatient. There's been one out of many studies that suggested maybe there could be some sort of outpatient benefit, maybe. So that's the idea, is that we don't like to give total certainty until we have certainty.
Yeah. So I'm looking at the list now. There are 89 drugs or combinations, and among the Ds are hydroxychloroquine, which is probably merited, I would say.
The most frequently used drug in the pandemic. It's still number one because there was so much use early on. It's the number one used drug in the pandemic.
Amazing. Favipiravir, which I think is an influenza antiviral, right? So I guess that didn't work.
Which was used extensively early on. Lopinavir, ritonavir was also used extensively early on.
And convalescent plasma is a D as well. But if you want to look at the other end, the A's, here, let's get to the top here. Maybe I could go home. Will that do it? Yeah, that did it. The A drugs include baricitinib. Remdesivir is also an A. Although
we basically, our algorithm right now requires you need to have at least one positive randomized controlled trial of over 1,000 patients to be in the running for an A. And then there's other things that can boost you up, but that's kind of the bare minimum, is 1,000 patient trial that achieves the primary endpoint. And both of those drugs had that.
There are only 4 A's actually: baricitinib, remdesivir, dexamethasone, and toci. Yeah,
and the reason that the monoclonals aren't yet an A is because we haven't separated into outpatient, inpatient. It will be an A. The monoclonals will be an outpatient A according to our algorithm. We just haven't updated the system yet, but they will not be an inpatient A because there's very little information on those monoclonals in the inpatient setting.
You have BCG on here as well.
Yeah, there's some, it's a B category, I think, because there's some, interesting, kind of, maybe early data that maybe giving BCG could boost the immune response.
I mean, it's only one trial with 30 patients, though. As you say, it's hard to conclude anything, right?
Yeah, those single trials of 30 patients that achieve the primary endpoint, those are the ones that really, that we really feel a responsibility to shepherd them forward, because all of us, myself included, would never say, like, give this because of a 30 patient trial, but someone's got to say, but let's move on to a larger trial and let's make sure that we don't just like let this fall to the wayside. And I mean, I think something that we're all well aware of is that the drugs that typically move forward are drugs that are on patent and have a shepherd, a company behind them that's saying, let's do more and more studies. And I think that's totally fine. I certainly have no problem whatsoever with that. But I do feel that for the vast majority of drugs where they're not on patent, they don't have a shepherd, someone's got to shepherd them. And so, we feel this responsibility to be the ones looking at these diamonds in the rough that no one's currently paying for, no one's doing the trials of, but there's something there that suggests they might be helpful. That's where we feel it's our responsibility to work with NIH and FDA to make sure someone studies them.
Yeah, I encourage everyone to go check it out. It's really informative. And you can click on each drug and you can see all the details. It's really good. And,
thank you.
Love it. It's really great, David. I just wanted to ask you a few more things that I gleaned from the book. First of all, is Red Bull really a good thing to do? It seems that you were really into it.
Yeah, it was because of this issue with fatigue. I mean, I was, early on, I was on, I was so tired that I was having to take, tons and tons of caffeine pills and caffeine drinks just to make it through like the day. I mean, as you know, I talked about in the book as well that I was on an OBGYN rotation, and I would go into a room, I would see a patient, and then I would go find an empty room in the hospital, and I would take a 7-minute nap. I would set my alarm for 7 minutes. I would wake up 7 minutes later, and I would go see another patient and go back. And that was the kind of fatigue that something was off. I mean, that's cytokine storm fatigue. That's not just like, tired med student fatigue.
I was just funny. We're always going back to the Red Bull. I guess it's pretty common in sleep-starved medical students.
It's very common in medical students. Yeah. I think Monster might have been out competing Red Bull back in those days, but Red Bull was definitely up there.
I love the story when you went to, I think it was Bethlehem, and you delivered a child, and the father goes, "This is not your first one, is it?" And you said, no, but it was your second, right?
It was. And I felt really conflicted about whether I should say that. I said, no, this isn't my first one. But yeah, But
it was the point you made, which is really good, there always has to be a first one, right?
Yeah, exactly. And of course, it came back so importantly for me with my disease and with sirolimus is that, there has to be a first patient to get sirolimus. With Castleman's, there has to be a first for everything, a first patient to get dexamethasone for COVID. And so, it's embracing those first and then tracking whether they actually work.
The other thing I really enjoyed that I want to point out is you were taking care of a patient with brain tumor, and you were giving him cognitive tests where in the first one he scored a 30 and he got so excited. And so you gave him a new one every day, and you said, it's not going in his chart. The only thing it does is make him feel better. And that's humanity, which is perfect, right? That's what you need. And I just wanted to ask you, I mean, they can't teach that in medical school, right?
You're right. I think it's hard to teach, but I had the best teacher in the world for that sort of thing. My mom was the most incredibly thoughtful and caring and just incredible person. And she had, as you know, passed away just a couple years before from the same type of cancer, also a glioblastoma. So when I interacted with this patient, I felt this connection to this patient and to his family, in particular, because I had gone through something so similar with my mom. And I think I really was able to channel, what would my mom do in this situation? And I think in medicine, we often, think about the patient in front of us in terms of their medical record number and, their medical history, and sometimes forget that they're a human and that they're, a mother or father or sister or brother or son or daughter. And so I think for me, having recently gone through that experience, just reminded me of that. And, I'll cherish those memories with that patient, forever.
Yeah, I think it's really important that medical students, doctors realize you have to be human in the end. You can have all the tech you want, but these are people and they have feelings, and not everyone does it. I mean, I've seen very cold physicians, unfortunately, right? Because you can't, it's hard to teach and it's not genetically in there.
No.
I don't know. Brianne, Kathy, anything else you'd like to chat about?
I just wondered what was next in terms of thinking about cytokine storms, but that's because I'm an immunologist who thinks about cytokine storms.
And I'll pipe up and say that I wanted to know if you think you're going to be doing more large-scale virus searches with respect to any other cytokine storm things.
Great question. So on the first question around our plans moving forward with cytokine storm, I think that the first step was defining it so we can all agree what is, what isn't it, and also disagree. But I think that the next step, once we have that definition, is really for us to come together, to collaborate, to bring together samples from various disease states, patients with COVID who aren't experiencing cytokine storm, patients with COVID who do have hyperinflammation, Castleman disease, and really actually start doing studies like kind of under the umbrella of cytokine storm. So many of us study, our silo. And I think that the next step, is to apply the tools that all of us are using. We're all doing single cell RNA sequencing. We're all doing serum proteomics. We're all doing the same sorts of things, but we're doing them in our silos. And so I think we got to come together and do these really massive, large scale studies, sample sizes in the thousands, to where we really can start to understand and the reason I really want to do it is not so much just to understand the biology, but really it's to understand, are there signatures for certain drugs where, forget the name you put on it, whether you call it Castleman cytokine storm or COVID cytokine storm, are there certain signatures that are suggestive that IL-6 blockade should be used or TNF alpha blockade should be used, so we can get kind of like disease name agnostic and really therapeutically focused, which is, I think, what we all want to get towards. So that's number one. Number two is that the step to connect what's going on biologically to what drugs are already FDA approved that could be repurposed, that's a big step right now. And that there's a big gap between, okay, I found a type 1 interferon signature and let's, inhibit type 1 interferon, with a JAK inhibitor or something like that. There's kind of a big step there. And I think that it's really important that that those of us that have kind of trekked along that challenging path, that we try to document and share and learn from one another, so that we as a medical system can get better at linking, okay, we found this in the lab, and oh, by the way, there's a drug that's already FDA approved that hits that thing. Let's do a great clinical trial to see whether it works or it doesn't work. And so that I think is really, really critical. And then the third, and to the question around future viral discovery and pathogen discovery work, I think it's really, really important. We've certainly all learned that there are, especially here with COVID, some of the manifestations that occur in the weeks and months after being infected with COVID, where the virus is gone, there's no evidence of it, yet you're experiencing MIS-C or you're experiencing, long COVID. These are our past. These are examples that maybe we've seen in other diseases. We just didn't know that 3 months before that patient showed up that they were infected with something else. And so I think it's really important that we do dig in further, although of course the challenge is when you got that biased specimen. If you don't get the sample until they show up 3 months later, maybe you don't have the evidence that you need. But I hope we can figure that out.
Yeah. All right. We've held you enough, David. Thank you so much. David,
I have so enjoyed this.
we have a good time here on TWiV. David Fajgenbaum from the University of Pennsylvania. Go buy his book, Chasing My Cure. It's fabulous. You'll love it. Thank you so much, David. It's been an inspiration. I really love what you're doing.
Yeah.
Well, thank you so much for having me. This was so wonderful.
Yeah, this was awesome. Thank you.
Great.
Yeah, and keep it up.
Can you
Man,
hear the massive storm here?
no, did you have a massive storm?
It's storming right
No, I
now. A
didn't.
lot. Yeah.
On Friday, we had one too, remember? On
Oh, I
TWiV.
could hear that in the recording. It
Yeah,
was
thunder
impressive.
and
Yeah.
pattering against my window. I don't think it's doing anything here, but I'm not far from you, so I'm surprised.
No, we have a lot of thunder.
Huh, cool.
Well, that was really great. And you said you met him on some Google calls or something?
Yeah, Google runs this Sunday evening conference of all mostly medical centers throughout the world, and so it's about COVID, and
Mhm.
I was invited on early to talk, and then I said, could I just come every time? And he said, yeah. So I just listened, and David talked a few times, and this database I thought was really cool.
Yeah.
But then, so when I invited him, he said yes. He said, here, have a copy of my book. I'm like, oh my gosh, I didn't know any of this. I mean, you would never know because he looks terrific. And I mean,
well, and we didn't even talk about one of the first things he started, which was this thing that led to his first book after his mother died. This network of people and things for people in that age group of college and very young adults who've lost a parent. And so there's a whole, and there started to be chapters at colleges and universities all around the country and stuff. So he's organized several things. So in addition to organizing the what is a cytokine storm and how are we going to classify that, it sounded like he had already cleaned up the mess of the different kinds of Castleman's disease, and that there was an agreement on that, and then now they have these criteria and so forth. But so he's had a lot of experience that he's been able to bring to bear.
Yeah.
For instance, on the coronavirus thing,
Yeah.
and just as he described, he thought, well, somebody should be doing this, and then a minute later, it's like, oh, we should do this.
Yeah, it's just the number of ideas he has and sort of the seeming amount of energy he has to pursue so many of them in such a smart way is awesome.
Yeah.
and plus, I think he's like the youngest faculty ever hired at the University of Pennsylvania. On top of it, it's amazing. I get tired thinking about all the things he's done. It's just incredible. It's great.
Yeah.
It's all good stuff too.
Yeah.
So, yeah, I that's why one of the reasons I like going to these meetings is that I meet people I would never meet otherwise, and I've always got an eye out for guests, right? And this lady who I've mentioned has a wonderful Zoom background. She used to work for the FDA. Man, she's great. She knows all kinds of drug stuff. And the thing is, during the talks, there's a little chat, and you can ask questions, and people chat back and forth, so they don't interrupt the speaker. It's really good. And I once asked a question, and she answered it right away. So she works for a company, but maybe she'd come on. She would be cool. That was really good. All right. Let's do a couple of email before we do our picks. And maybe, Brianne, you would like to take that first one.
Sure. Pamela writes, hello. As one of the co-signers of the letter to science asking for increased investigation into the origins of SARS-CoV-2, I want to make 2 points. First, thanks very much for the 2 episodes of TWiV when you met with the WHO committee and with Robert Gary. These episodes are doing a great service for all to clarify these issues. Second, as a co-signer on the letter to science, I'd like to explain my motivation for adding my name. Namely, I thought the letter would have the effect of prompting more funding for searching for natural viruses in animal reservoirs, which I personally have always assumed represent the origin of SARS-CoV-2 infections in humans. Perhaps naively, I did not anticipate that the letter would be used to promote the lab origin hypothesis. Looking back on the wording of the letter, however, I now think that I should have realized this would happen and should have been more proactive, either not signed the letter at all or else requested more wording changes to make my position clear. Thanks for everything you're doing on TWiV. I listened to every episode and have learned a lot. Best regards. And this is from Pamela Bjorkman, who is a pretty famous immunologist, virologist.
Yeah, it's cool. Was that thunder I just heard?
I think probably,
wow,
yes.
Cool. It's propitious. It's the right moment.
Yes.
Now, I appreciate it. This just came in, and I'm very happy. So Pamela's also on these calls that I've met David on, and I thought we have to get her on as well. And, this is the science letter that came out a few weeks ago that a little bit puzzling. They called for an investigation, which, well, that's what the WHO committee, which has got a lot of good people on it, is doing. So I'm glad she wrote this. It's really good.
It helps clarify. And I imagine she's not the only one who has that feeling of, I signed this letter because I want there to be more investigation into the natural infection origins.
Yeah, I also recently saw a story about the situation where someone called part of the SARS-CoV-2 genome a smoking gun, and that being a comment that they have also noted they were sort of taken out of context and misquoted. And so I think that some of these things are perhaps a little bit of that, as she mentions here. Yeah.
I don't really understand the purpose of that letter, especially in light of the WHO committee, but so be it.
Yeah, I think people just maybe did not know. I mean, if our episode 760 with those three people could have come out, before that letter was written, maybe they wouldn't have written the letter.
I also think that a lot of people haven't read the actual WHO report, which we did on TWiV a while ago and went through it. It's quite good. And if you just hear what you're hearing in mainstream news, then you'd have a different opinion. So,
right.
Kathy, can you take the next one?
Yes. Dirk writes, Dear virologists, summer is starting in Belgium. We notice that the rain gets warmer. The major news related to virology is that a terrorist is on the loose in Belgium. He is military personnel and was recently listed as potentially dangerous extremist, but this was not followed up upon. So one evening, he left work with a machine gun, a pistol, 4 rocket launchers, and ammunition. He wrote farewell letters in which he blamed the government and the experts for the semi-lockdowns, and he specifically threatened the Anthony Fauci of Belgium, Professor Mark van Ranst. Professor van Ranst is not only a virologist, but he also fiercely criticizes any form of discrimination, so is therefore a favorite pick of right-wing extremists. No people have been hurt yet, but the terrorist did try to booby-trap his car and was spotted near the house of Professor Van Ranst just before disappearing. And he gave a link. Anyway, I hope he gives up or they find him soon, as should be doing other things these days. Otherwise, vaccination is going okay. Numbers are dropping. Hopes are high. If everyone acts a bit normal, we will get there. Thanks for the podcast. Regards, Dirk. And so I can't tell from this link when that article was written, but I looked, this has been in the show notes for a little while. There was an update. The terrorist is still on the loose as of May 31st and as of June 5th. So there's an article in politico.eu. The true shock factor of the case is that tens of thousands of people have rallied to support him, and that far-right Flemish nationalists from the Dutch-speaking North are styling him as a symbol of their deeper political grievances. So once again, we have politics mixing with science.
Wow.
Well, I hope he's apprehended soon.
Yeah, just terrible. Thank you, Dirk. Dirk is a virologist at the Rig Institute.
But other than his writing in, I had not heard or seen anything about this story, which sounds like
you didn't?
Really
it's on BBC News, yeah.
Yeah, but it's kind of terrorizing Belgium. And so this Professor Van Ranst, has protection. Other people have now received protection as well. It's really scary.
Mm-hmm. Christy writes, Dear TWivers, a quick one for the listener whose Catholic grandmother is concerned about taking a vaccine for ethical reasons. Perhaps that was because of the use of HEC293 cells, etc., which we talked about. Perhaps the grandmother can consider the ethics of not getting vaccinated. For those individuals who can't get vaccinated for medical reasons, and for those individuals who have medical conditions that reduce the effectiveness of the vaccine for them, don't we all have a responsibility to get vaccinated to protect them? I'm specifically thinking of a friend who, due to the drugs she is taking as a result of her kidney transplant, has been told that the likelihood the vaccine will have antibody-building effects for her are about 17%. Thank you all for the time and energy you put in. It matters, Christy. I agree, Christy. I always think that you have to think of others, but not everyone thinks that way. Not everyone is altruistic. I remember when it was time to give our kids polio vaccine, and at the time, you had a choice between Sabin and Salk, oral polio vaccine and inactivated. And I remember for our first child, the doc, she knew I was a virologist. She said, I assume you want the Sabin vaccine, right? I said, yeah. He said, because it protects others. It inhibits transmission. And I knew that, there was a 1 in 1.5 million chance that my son could get paralyzed from it, but I thought it was important to do it for the population. Now, not everyone thinks that way. You're a human. You have a right to think as you wish, but, I think we have to think of each other more, and that would really, and my pick is actually related to this. Let's do one more round. Brianne?
All right. Joshua writes, turns out that Michael R. Gordon, the lead author on that Wall Street Journal piece that has mainstreamed the rampant conjecture surrounding the origins of SARS-CoV-2, was one of the two authors who wrote the infamous September 8th, 2002 article in the New York Times that mainstreamed the false allegations that Saddam Hussein had weapons of mass destruction in Iraq. Link to an article about the whole thing, and he gives a link. These claims of Iraqi WMDs were central to the U.S. justification for invading that same country in 2003. These allegations, however, were known to be false by the executive branch, and were actively spread by then VP Dick Cheney. The fact that this charlatan, I'm referring to Mr. Gordon here, has a job as a journalist after 2003 is ridiculous. The fact that his work is again being widely cited at all is a sick joke. And the fact that his evidence-free writing is being used once again to further stoke political animosity in this country is nothing short of a goddamn shame. And worst of all, not a peep about this author's history of shameful reporting in the mainstream media in the US. Instead, his work is given the greatest platform imaginable, while the actual science on the origins of the virus is thrown to the wayside. You have to go to outlets like the Chinese state media or the link given above to even see this connection being made. I'm at a loss for words. Sincerely, Josh, an incredibly pissed off ecohydrology technician in southwestern Georgia. P.S. In my last note, I erroneously said that in 2003, the U.S. invaded Afghanistan. That was false. In 2003, the U.S. invaded Iraq. We had invaded Afghanistan by the end of 2001. I apologize for the error, but unlike Mr. Gordon, my oopsie isn't going to start a war.
Now, someone's going to write and say, how dare you agree with whatever Josh is saying? We're just reading it, folks, okay? If you think it's not correct, that's your thing. We're reading people's letters. Doesn't mean we endorse them, especially in this case. It sounds pretty nasty that he did this. And then, this Wall Street Journal article about the SARS-CoV-2 origins is just full of nonsense. The three people at the Wuhan Institute who supposedly got sick, they didn't have COVID. Koopman said they didn't have COVID. The miners, they didn't have COVID years ago in that cave in Yunnan. None of it's true. It's garbage. And yet there have been multiple op-eds in the Wall Street Journal now that cite that. And then there was one the other day that cite the State Department document. Remember that? We covered it last year, which is full of garbage. And they say this is like compelling. I don't get it.
Yeah.
I don't get it.
I don't get it. I don't really get the motivation for it either.
The science is ignored, right?
Bad.
The science is ignored. Just let's look at the rumors. It's crazy. I just hope it passes as quickly as possible because very frustrating. Kathy.
H writes, it's hot here, just hot here in Sonoma County. So I will be refusing to look at the thermometer so it doesn't get to my head more than it already has. I have one thought and a question for you all. Please imagine the following using Dixon's voice. I'm most like him amongst you. Well, just a printmaker, however. A thought. I dabble, in a most amateurish way, in cancer cure stocks. So without naming names, I was reading some obscure sets of agreements between an immune oncology company, less than $10 billion market cap, and a more basic cell therapy company, less than $10 million. The latter will later affirm that it will become a subsidiary of the former. What caught my eyes was the date of this particular agreement regarding the royalty that the larger company, using the smaller company's technologies, will be paying depending on the future milestone achievements of the larger company, January 1st, 2020. In the agreement, it references SARS-CoV-2 alongside vaccines as one of the future goals. As we are painfully aware, here are some pertinent items in the SARS-CoV-2 timeline. The first patient was sort of identified in late December of 2019. We later think we have some cases earlier, but that's not important here. A bunch of unidentified pneumonia in Wuhan area around the same time. January 9th, WHO announces mysterious coronavirus-related pneumonia in Wuhan, China. The virus was first sequenced sometime between the 10th to the 12th of January 2020. January 31st, WHO issues global health emergency. I, for myself, ordered a bunch of N95 masks in late January 2020 after seeing things are starting to get out of control in Hong Kong. This was way before issues arose with the masks as we know today. Now, given that, were these guys who worked out the agreement between the two companies just super smart and or lucky? I am probably the least conspiracy theory type that I know of, so I'm not going there. But it seems like someone really dropped the ball, don't you think? To get us to what we've gone through in the past 1.5 years? Clearly, there were some people with one of our most innate of all motives, money, to draw up this document weeks before you guys even talked about SARS-CoV-2. It says there were a bunch of lawyers working on the agreement on the Monday and Tuesday before the new year on Wednesday. Oh, wait, you guys need to invite them all for TWiV. LOL. And a question. You dealt briefly on this matter a few weeks back, but I would like to know more in details if I may. How does it work that some vaccines can elicit greater or better immune responses than the actual infections by the pathogens themselves? I would like to know the mechanism of that. I have a feeling it connects to tumor-associated antigen that I read with oncology stuff. It gives a link. Are all the items that are not necessary for the public consumption as I have a vested financial interest? Oh, that's an asterisk relating to something up above in his letter. So if you go online and read these letters, you can connect that footnote. Great. Thanks as always, H.
Yeah, the link is to this agreement that he's talking about between the two companies.
Got it.
So, Brianne, tell him the answer to that question, please.
So when you are getting infected with the entire pathogen, there are some proteins that can inhibit immune responses, in addition to the ones that you are trying to make the immune response to, in addition to those antigens. When you are getting the vaccine, you are going to not have those inhibitory proteins. You also could potentially, with the vaccine, have differences in terms of amounts of antigen or delivery location of antigen or things like that that could give you a better immune response.
Okay. I think these guys got lucky. They made a gamble. They said, well, there was a SARS-1, and maybe this will be bigger, and there's no downside to the agreement. And they lucked out because I think January 1, nobody knew where this was headed. I mean, the sequence was, the virus was first sequenced between the 10th and 12th, but isn't that when they published it or announced it or released it, right?
Yeah, it's
Right.
around then. And I feel like they said that it was within a day or two that they started working on the mRNA-based vaccines.
Yeah.
But there's something fishy about this too, though, because they're calling it COVID and SARS-CoV-2.
Yeah, it was too soon. They didn't give it that name.
It was novel coronavirus 2019.
Yeah, for a while, right? Yeah.
And then they named the virus and the disease sometime
Yeah,
later.
and they named the, well into January. It was while I was teaching my virology course during the semester that name change happened.
Yeah.
So maybe this document is backdated.
Could be.
Yeah.
All right, one more from Brenda. Honored TWiVers, I'm writing to you in a state of anger and distress. I alternated between screaming with rage and crying with frustration. After all your good work uncovering the investigations into the origin of SARS-CoV-2, The Guardian, which I had till now regarded as the last man standing in decent reporting, puts out this. Wuhan coronavirus lab leak or, yeah, right.
Yeah.
What really had me seething was this caption to a photograph of masked guards at the Wuhan Institute. And what is this?
The caption says, "My own complacency on the matter was dynamited by the lab leak essay that ran in the Bulletin of Atomic Scientists earlier this month." But it doesn't say who that's quoting, unless it's the author.
I think it's the author of the piece
of the op-ed.
that the op-ed said, was thinking they didn't really think this was important until they read the Bulletin of Atomic Scientists.
Yeah, which is the Nicholas Wade article, right?
Yep.
Right.
Well, Brenda, we share your anger and distress, and I don't know what to do about it. But yes, everyone is ignoring the science. And Nicholas Wade and others, the Wall Street Journal, have really amplified this from nothing. They really have nothing.
Yeah, we're starting to hope that some other more reasoned things are coming out. For instance, there was an article in the New York Times by a business columnist, and I'm trying to pull up his name, that is starting to give some pushback. The title of this column is "The Lab Leak Origin Claim for COVID-19 is in the news, but it's still fact-free."
Yeah, good.
So his name is Michael Hiltzik, Hiltzik, if somebody wants to look it up.
It is fact-free.
Yeah.
All right, time for some picks. Rhiannon, what do you have for us?
So I have an article that I came across recently that is actually about giraffes. And it is called "How Giraffes Beat High Blood Pressure." And it talks about the fact that giraffes, in fact, have incredibly high blood pressure, about twice what humans have. And biologists quest to figure out how that is okay and how it is that giraffes can actually survive with this higher blood pressure. And so what kind of cardiovascular or other physiologic alterations do they have that allow that to work, and sort of how we might be able to think about some other aspects of our own biology based on that knowledge.
Cool.
It's very good.
Giraffes are fairly skittish animals. I remember hearing about when there were wildfires near the San Diego Zoo Safari Park. That's when I first learned that they're really skittish, and they had to very carefully take care of them.
They're amazing.
Yeah.
I remember when we used to have a zoo here in town, little town of 24,000 people. It had a zoo. It closed years ago and they actually had non-human primates and giraffes.
Wow.
They were closed because they weren't taking good care of them. But we went once, and it was called the Terry Lou Zoo. Yeah, that's right. And so Aiden was like a little guy, really short, young, and the giraffe. The fence was low enough so the giraffe put his or her head over, and then the kids were all feeding them peanuts, right? And the kids would put the peanuts on their palm, and the giraffe would lick it off. And Aiden was giggling because the tongue was so soft, right? And he looked at me giggling, and this giraffe was just gently licking it. And they would go to the next kid with, "Oh, so cool." I mean, very gentle and calm.
Oh, that's awesome.
It must have been used to it, right? They're amazing. Yeah, I love giraffes. Kathy, what do you have for us?
I picked something. Speaking of BBC, this is an optical illusion of an orangutan that wins an award. It's a mind-bending photograph where the sky is reflecting in water. And basically, you start looking at this picture, and then it takes a while to figure out what you're looking at and how this picture works. I had to go away and come back and look at it again, and then I was able to figure it out. But I'm going to send it to Dixon, too, because it's the kind of thing that he would enjoy seeing, I'm sure. And it leads to some other photographs in this judging or contest thing.
It's beautiful.
But this one is amazing.
Beautiful.
That is very cool. It does take a while to
Yeah.
sort of look at and figure out exactly what's up.
Yeah, you're looking at the orangutan climbing up toward you, but you see the sky and the tops of the trees behind it. It's very weird.
Oh, wow. Now I'm looking at some of the other
The
ones.
others are great too. Other
This
great.
one,
Yeah.
the night sky.
Yeah.
Other really good ones. Nice. Very
Yeah.
nice. Brienne, I just got the thunder here. I just heard it.
We've been having it for a while.
God, it took that long to get here. How about that? All right. So I want to pick two quotes from Chasing My Cure. I mean, I could pick the book, but I'll put a link to Chasing My Cure. You should buy it and read it. It's really good. It's really well written, too. He's a good writer.
Mm-hmm.
I don't know how he remembered all that stuff, especially when he was sick, right? Because you tend to be in a fog. And then what he had, man, was really horrible.
Yeah.
Anyway, so he has a quote from John Paul II. And it reads, "Dear young people, whether you are believers or not, accept the call to be virtuous. This means being strong within, having a big heart, being rich in the highest sentiments, bold in the truth, courageous in freedom, constant in responsibility, generous in love, and invincible in hope." And I think that's a great quote, right? We should all be all of these things, at least strive. Okay, and then a quote from David. He, at some point, said, "Never stop questioning and always follow the data, folks. It's all about the data in science, and seems to be not being paid attention to by many people with respect to the origin of SARS-CoV-2. Always follow the data. That's what scientists do." So I think that's a great. Those are two great quotes. I just wanted to pick them, to highlight them, and ask that we follow them. So that's Chasing the Cure by David Fajgenbaum. And I remember when his name came up and someone pronounced it, I said, wow, I never would have pronounced it, because it's spelled Fajgenbaum. All right, that'll do it for TWiV 766. The show notes are at microbe.tv/TWiV. Send us your questions and comments, twiv@microbe.tv. If you like what we do, consider supporting us, microbe.tv/contribute. Kathy Spindler's at the University of Michigan in Ann Arbor. Thanks, Kathy.
Thanks. This is a lot of fun.
Brienne Barker's at Drew University, BioProf Barker on Twitter. Thanks, Brienne.
Thanks. It was great to be here. He was just fascinating.
Yeah, it's terrific. Really good.
Thanks for inviting him.
Yeah, my pleasure. I'm Vincent Racaniello. You can find me at virology.blog. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV, and Ronald Jenkins for the music. This episode of TWiV was recorded, edited, and posted by me, Vincent Racaniello. You've been listening to This Week in Virology. Thanks for joining us. We'll be back soon. Another TWiV is viral.